Okay, just a quick definition of what a metacommunity is. A metacommunity consists of local communities that can exchange individuals between them, different rates, different dispersal rates. Now, if we're talking about birds, dispersal can be a bird that flies from one community to another. But the terrestrial plants have seeds that can be carried away with the winds. But when we're talking about aquatic metacommunities, these local patches have to be connected, hydraulically connected, in order for the aquatic organisms to be able to move between them. So, when we consider fragmentation in aquatic systems, fragmentation can be anthropogenic, man-made structures such as dams in rivers or dredging activities that can affect connectivity, but can also be natural, for example in coastal systems, different lagoons or rock pools. And in this system we can have nutrient loading due to terrestrial runoffs or due to wave activity, and the different nutrient loadings can act to create heterogeneous systems and that can have different species compositions. For example, you can see the different colors in the different pools, so that means we have different species there. Now, these systems can be fragmented, but they're not forever fragmented. They can be periodically connected through waves, for example. So we believe that it is important to try to study together these two activities. We know that biodiversity is very important because it can benefit ecosystem functions and processes such as productivity and stability, and then in turn benefit ecosystem goods and services. Now, the aim of this study is to affect the effects of both fragmentation and connectivity in these systems, and we investigate species composition therein, the structure of the community, and the function of the community, such as productivity and stability. And we're using an experimental approach, laboratory microcosms, with natural phytoplankton assemblages. So what we did, we took water from the coastal area of the Aegean, near the island of Lesbos in Greece, and during the succession of these communities, we measured about 100 species, phytoplankton species in there. And then we fragmented them into different local assemblages. In order to create heterogeneity in there, we used different nutrient pulsing frequencies of 1 day, 3 day, and 6 day. And nutrient pulses in phytoplankton can be regarded as different disturbances. So usually they create different species compositions. And then we monitored all these cultures for about 3 months. We started from, now I don't think you can see a lot there, but we have an initial species pool with many species there, many functionally different species. And after the course of the time, we reached what we call a steady state. That is that our cultures have the same species composition and pretty much the same relative abundance through a period of approximately 20 days. And then, as you can see, when we reach steady state, our different pulsing frequencies have different species richness, with our less frequent pulse having more species, and with the most frequent pulse having greater evenness. But also, we saw that with species richness, we also have an increase in functional diversity. We have only one functional group in the 1 day, in the most frequent pulsing frequency, and up to 3 functional groups when pulsing frequency was lower. And then what we did was we took these isolated assemblages we created, and then we connected them under different connectivity levels to assess also the effect of the level and the severity of connectivity. So what we saw was regarding species richness. Intermediate and low connectivity have increased species richness in its local system compared to the isolated assemblages. Productivity was also increased at the connected systems, at the metacommunities, compared again to the isolated assemblages. The coefficient of variation of productivity, which is a common measure of stability, was also decreased. That means that the connected assemblages were more stable. And then we also measured a community synchrony, which actually shows the higher the synchrony of the community means that the species oscillate synchronously, and we have a probability of having less species together, so higher smaller stability too. But as you can see, synchrony doesn't really change, and that's reflecting to stability here. Now, this is a look inside the cultures. Here we have the monitoring of during a month of the isolated cultures and of the different metacommunities. As you can see, our most productive species was a species here, that's an Itzia species, a diatom, that actually after dispersal, after connectivity, dominated across all the metacommunities, across all the local systems. So, but this species was actually in our first species pool, in our initial species pool, but could dominate only here. So our conclusions, to sum up, fragmentation of the same species pool under different nutrient regimes can result in different species composition and structure. Our less frequent nutrient pulses can increase both species richness and functional diversity. Our connected patches have higher species richness, productivity, and stability compared to isolated patches. But there is only one very productive species that dominated across the metacommunity and resulted into this pattern we observed. And of course, the conditions during fragmentations were the ones that enabled the dominance of this species in just one patch. And it is important, we think, it shows the importance of looking at the effects of both fragmentation and connectivity together. Well, we all know that climate change can influence habitat fragmentation and connectivity. This is an example from Santa Lucia Lagoon in South Africa, where severe droughts at some years and decreased a lot biodiversity. And we do know that our results show that connectivity is very important. But this dominance of really one species, is it a risk of eutrophication phenomena, of algal blooms? So we really want to check this and try to generalize our findings beyond specific species and specific conditions in order to give a solid case for coastal management. Thank you very much for your attention.
Thanks, Eva. It's really interesting to see how the functionality of these algal communities is impacted by not only spatial